You're listening to the MER-Mates podcast, the Marine Science Research podcast.
When people think of scientists, nerdy might come to mind. But what about sexy?
Today's guest proves that they can be both. Meet the man who came up with the term sexy scientist and works with the science of sex differentiation in fish oocytes and testis.
But that's not all. We will also explore some of the thrilling stories from past and present scientific expeditions, where fieldwork gets tough and the scientists even tougher. Tune in for a new episode and may the science be with you.
Today we have with us a full professor in cell biology. His research also is focused on two main interrelated lines: cell and molecular biology of fish sex differentiation and determination, and applications of cell biology in toxicology. His PhD was done with the University of the Basque Country, characterization and biogenesis of peroxisomes in mussels. He's carried out research stays in Wales, Netherlands, Germany, Scotland, Belgium, and there's lots more that we hope we can get to talk about today. We have with us here Ibon Cancio. Thank you for being with us, and you're very welcome.
It's a pleasure.
Ibon, the first question is compulsory for us. We've had the pleasure to attend some of your lectures, and we have a huge doubt that didn't let us sleep the past few days. Do you consider us sexy?
Yeah.
We do consider you sexy.
I do not consider you both sexy, yeah, but science is sexy.
There you go.
And I think that most of the people coming to science, they do at a certain point in their lives, they decided that they wanted to dedicate their lives to science. And I'm fed up about boring lectures and people just drowsing in the lecture theaters. If they were coming here to receive something, they should be given that something. And I did try to do that.
You've also clarified the concept of being sexy. We do refer to sexy scientists, although both in our case maybe in here. How did you come up with the concept of sexy scientists?
It's this idea of something being attractive, no? And I think that science, it's attractive. And in coming to lecture, I mean, you are selling a product, and you have to sell that product under a very shiny and bright envelope. And that's how I do it. I use the word sexy, and suddenly I see that the eyes in the audience spark just by saying the word. I mean, it's not only with the students. It also happens with grown-ups and rocket science making scientists. When you mention the word sexy, clack, everybody's happy about what you are going to be telling. And I like people being happy when I'm talking in front of them.
Following with the sexy word, what sexy science are you currently working with?
Okay, that's another reason for me to use the word sexy, because I do work in sex differentiation, not so much in sex determination. My main focus of research is the oocyte in fish, and I do work in identifying different molecules that identify oocytes along development within the ovary of fish, and also within the testis of fish, because under certain circumstances, mainly under exposure to compounds that are similar to the estrogenic hormone, there are cases of feminizations in males, so they begin to develop oocytes within the testis. So my markers of differentiation of oocytes serve to identify intersex individuals in fish, and that's for me the very interesting thing of my work. I do work at the molecular level, but I am able to identify changes at the molecular level to a phenotype. And this phenotype is the differentiation of sex. It's very nice when you have a molecule that is telling you this is female, or you have a molecule that is telling you this is male, or you have a molecule that is saying, okay, you have a male which is becoming female. Yeah? So that's my work.
Following with this, we recently had the pleasure to talk with Belen, and we mentioned some of the emerging pollutants that they can act as hormone disruptors, and they can induce or provoke intersex in some species. And I guess you have worked recently on them. What can we learn from your experience?
Okay, my first experience with these compounds, we call them emerging compounds. Yeah, emerging compounds they are called emerging compounds because our analytical chemistry capacities are improving a lot, and we are able to measure really, really tiny concentrations now in water, something that we were not able to do before, and that's why we call them emerging. The problems with xenoestrogenic compounds, those compounds that are similar to the female reproductive hormone, began to arise sometime in the 80s. And in the 80s, it became apparent that there was problems of feminization of fish in some river areas. Mainly this came out in the UK. And it was then when they identified that the main causing or driver chemical could be ethinyl estradiol, which is one of the main components within the contraceptive pill. So, I mean, in the 60s, the contraceptive pill was first commercialized. That brought, as it used to be sold at the time, freedom for females, for women. And what it happened is that free women, when they take the contraceptive pill, they make PP as any human living organism. And in making PP, they are throwing ethinyl estradiol into the water, and then we have organisms swimming in between these chemicals that are telling them that they are females. But there are many chemical compounds that have got somehow a similar structure to estradiol, the female hormone. Ethinyl estradiol is a very potent estrogen, but there are other chemical compounds that not being so effective as ethinyl estradiol and estradiol itself, they can react with the estrogen receptor and then trigger molecular pathways that result in differentiation of gametes into oocytes. Then there are other compounds that, not being estrogenic, are anti-androgenic, so then they have the same effect. And on top of that, we have compounds that are androgenic or anti-androgenic. So we have a complete pharmacy of chemical compounds out there that are sending different kinds of messages to the organisms living out there. And that's what I study. I study these messages and how these messages are disrupted.
And I guess that from the 60s to currently, there's been an increase of these compounds in our waters.
Yeah, I mean, the human brain is something amazing. Analytic and chemists are very intelligent people, and they are producing many chemical compounds that do our lives easier and better. No? And in doing so, we are unaware many times, or we have been in the past unaware of which could be the side effects of these chemical compounds, and we have been happily using them. And after happily using them, they come to the environment. And when they come to the environment, they come to our water bodies. And when they come to the water bodies, they come to the oceans. And they have effects on living organisms. And to tell the truth, we only have are beginning to understand which is the possible effect of these compounds, mainly on vertebrate organisms. I do work on fish, which are vertebrates and have got a liver, as you and myself do have a liver. And we know many of the molecular pathways that are triggered in these organisms. But vertebrates are not the only organisms out there. I mean, the sea is not only formed by dolphins and whales. There are plenty of organisms that are, for instance, invertebrate organisms. And we are only beginning to understand which are the molecular pathways that are regulated and that regulate, for instance, sex differentiation. We know nearly nothing about how sex is regulated or how sex differentiation happens in many of these organisms, and we do not have a clue of what these chemical compounds are doing to them.
I would like to know, are these problems found only in transitional waters, like it can be, for example, estuaries that connect our wastewater treatment plants with the ocean? Or is it something that we can find maybe in the middle of the ocean, a problem this type?
It's like with any other chemical compound. I mean, concentrations are important. Okay, when it comes to estrogenic compounds, it's quite funny because most of the xenoestrogenic effects, for instance, happen when the chemicals are at very low concentrations. That's when these chemicals can interact with the estrogen receptor. Then at a given higher concentration, they have other kinds of effects. But most of the effects, most of the cases of intersex that it have been identified in fish species, they have occurred in riparian waters, in fresh waters, mainly linked to the discharges of wastewater treatment plants, because when you make PP, our PP goes to the wastewater treatment plant, and the wastewater treatment plants are not able to effectively clean all these chemical compounds there. And then when they discharge in focus places, they focus some concentrations of these chemical compounds. Then we have been able to identify intersex individuals in fish from transitional waters, estuaries, as you have said, because there are also wastewater treatment plants that are discharging here, and we have very nice examples here in the Basque Country. Then when you move to the open sea, I mean, all these chemical compounds get very diluted, and there are very few examples of intersex condition happening or intersex phenomena occurring in marine waters. They have been described. They have been described in cod, for instance, in Norway. They could be going to fjords where they could be exposed to certain chemical compounds. Then it's also very important in which moment during their life period these organisms are exposed to the chemical compounds. So, for instance, when you think about coastal species like flatfish, flounder, for instance, or Dicentrachus labrax, sea bass. Those normally the larval stages, they develop in estuarine waters, and then they migrate to the ocean. So during their very important time windows during their development, if they are exposed to these chemical compounds, this can have effect on their sexual differentiation. And then they could be migrating to the sea where they are not going to be exposed anymore, but they could have already been imprinted by the chemicals they were exposed during their larval period.
Moving on, you've recently been moving around quite a bit.
Yeah.
In the past few weeks, you've been in Nantes, in Barcelona, Brest, and you still have to get to Brussels and Madrid. So you've got an extremely busy schedule. We're thankful for the fact that you found some time for this podcast. But we would like to know, what is it that you've been doing in all of these cities? Because I guess it wasn't tourism.
No, it wasn't. I wouldn't go for tourism to Brest. I would go to Nantes for tourism, and even to Barcelona. But okay, in Nantes, I was in a conference. It was a congress, Primo, Pollution Responses in Marine Organisms. It's a conference that is organized every 2 years. This has been the 22nd edition. It's a very nice congress. It's around the size that I like. It's the size of a congress in which you know most of the people and you can touch them and they touch you because we are friends. It's a very, very nice congress. And I was presenting some work by one of my PhD students. I should say a very sexy work. And it was nice. I had even the opportunity to use Ennio Morricone in my conference. And it was funny because we were invited to organize the 24th edition of Primo. It will happen in Bilbao in 2028, after it happens in 2026 in Hamilton, next to the Niagara Falls. So that's what I was doing in Nantes. In Barcelona, I was invited to deliver a talk in the CSIC, in the Institute for Marine Science, and to meet with the directory board of the institution, because they are willing to, or they want to have first-hand information on what's EMBRC. EMBRC is the European Marine Biological Resources Center, which is a research infrastructure, a European research infrastructure, in which Spain participates, and I am the director of EMBRC Spain. So I was there explaining what means being EMBRC, what it would mean for them becoming a member of EMBRC, and which would be the steps that they would have to follow. Then in Brest, I was in a meeting of an Interreg project that we have, in Interreg Atlantic area project that we have, in which we are trying to develop or initiate small research projects mainly for companies, for members outside academia, also public, or city halls or authorities that are willing to initiate small projects to launch different products or different projects. I mean, we have the money, we have seed money that we could be giving to them. And we are doing that in a trans-regional way. So we have partners from the Canary Islands, from north of Portugal, from Galicia, from the Basque Country, from Brittany and Ireland. And these seed projects are going to be run in between two regions to show that there are regional problems that could be tackled in the world of marine biotechnology with trans-regional collaboration. That's what I did in Brest. Next week, where do I have to go? I have to go to Brussels, So sexy things happen in the lab, but important things happen in Brussels. And there is a meeting organized by the European Parliament. DG Mare and DG Regio are trying to implement this idea that I have had for a long time of trans-regional collaboration in marine biotechnology. If marine biotechnology is going to develop in Europe, this is going to be a mosaic that is created from the regions that are connected to the sea. So there is not going to be a marine biotechnology in Europe that is going to be created from Brussels, from Munich, from Paris. It's going to be created in the different regions. And if it's going to be created in the different regions, we have to find ways in which we are not going to compete. Okay, we have to compete, but mainly we are going to collaborate. And we have a meeting on that next week. And my travel to Madrid is because the ministry is there.
Interesting timing of the meeting in Brussels, very close to the European elections.
You have to go after the elections. Will someone listen to you?
Yeah, I don't know. We will have nice conversations because the right-wing tide that it's coming to us, it gives for a good conversation.
Since we're talking about moving around for science, we would like to retrace back to the past and the birth of the marine research with the very first expeditions that happened in the last century. What is, in your opinion, one of the most relevant?
Okay, you always know that my students, whenever Darwin is in the room, have to stand up. And I mean, that's compulsory. Our reason as a biologist, my reason to do science, I think it's the evolution theory. And of course, the most important expedition, even when it was not such, I mean, it was a scientific expedition to some extent, but it was not the main focus, is the Beagle, the expedition of the Beagle. And the very important thing about the Beagle is that Darwin was on board of Beagle. So for me, top of the charts is Beagle. Then when it comes to the importance in science, I would say that the Challenger expedition is second best expedition. Should be maybe it's the most important, because you must consider that the beginning of exploratory or expedition voyages, it's something that it belongs to the British Empire. So it was during the period of the before the period of the Enlightenment. I mean, the big British Empire realized that science was not an expense; it was an investment. Science or knowledge gave the power. So they were always in their army vessels. They were enrolling naturalists or scientists. At the time, they were called naturalists, because most of the products that you could be obtaining were not nature based. We were traveling to other countries to find the spices or to find biological products, plants, whatsoever. And the thing is that in those vessels, they were always army vessels. Yeah, the Beagle was an army vessel; it had 10 cannons. But then in the Challenger expedition, this was a 100% scientific expedition. They deployed the Challenger from all the canyons because they needed the space for all the scientific equipment. and all the people traveling there were scientists. And the idea was to lay a telegraphic cable from Europe to America, and they circumnavigated the planet, dragging the bottoms of the sea for all along their trip and collecting different samples. That's how they discovered the deepest point in the ocean. It's the Challenger Deep in the how is it called?
Mariana Trench.
The Mariana Trench. That was a discovery of the Challenger expedition, and what they discovered was living organisms at very, very, very deep waters, because at that time it was supposed that living organisms were not able to survive under a certain depth. I think it was 600 or 800 meters. And suddenly they discovered that in the whole of the depth of the ocean there could be living organisms, and they came back with 4,700 different living organisms. And for me, the most amazing thing is that they came back in 1876. When they came back from Uruguay, they met another circumnavigating expedition. It was a German expedition, Lagassel, and they decided to came back through two different transects. So they were not competing. They decided to collaborate and come back through different transects. So for me, it's the first moment in history in which different nations decide to collaborate to do science. But it's not only that. Challenger, when it came back to the UK, to Scotland, they established an office in Edinburgh where all these 4,700 new living organisms were kept. And they decided, okay, if we need to study these organisms, we need the best brains to study them. And they didn't keep them for being studied by the professors in the University of Edinburgh. They called all the most important researchers all around the world, and they were specialists in different taxa that had access to these organisms. And there were scientists from Germany, from the United States, from Denmark, from Sweden, from Italy, that were called for this. For me, with this research by the sea, transnational cooperation began. And that's what is science, transnational cooperation, nothing else.
That's the power of science, no?
Yeah.
You've mentioned 2 or 3 expeditions, like the top for you. Now we can do the same for historical researchers. I guess your favorite, we can guess who is your favorite, but could you tell us someone who's not that famous or which might be a bit hidden during the history?
There is one for me that it's very important and very relevant. It's somehow connected to my field of research. He was an embryologist, Ernest Everett Just. He was a Black American scientist, one of the first to become a doctor in the United States in the 19th century. He obtained his PhD from the University of Chicago, and he did most of his research during summertime in Boots Hole, in the marine station there, in the Marine Biological Lab in Boots Hole. It has been traditionally, since it opened its doors in 1888, a place that has been like a shelter place for researchers of different universities in the United States. So University of Chicago, University of Harvard, many different places. They traditionally had traveled there in summertime to teach and to do research. And that was the case of Ernest Everett Just, who was only allowed to give lectures in universities for Black people. So he had to teach a lot, and he couldn't do any research in his free time. Being a Black scientist, he was treated in Boots Hole as a technician because he was technically very well suited, very well trained. And he didn't like too much how it was treated. He was visiting Boots Hole for 15 years, 1 year after the other. And he's the father of 2 of the main mechanisms that we know in fertilization, which is the fast and the slow block of polyspermy. You know that it's only one sperm and one oocyte. Yeah? So when one sperm is successful, the oocyte has to close any possibilities for any second sperm to come in. And there are two mechanisms, the fast and the slow block of polyspermy, that he described in different marine invertebrates in the ocean. Okay, I have brought you for the 15 years of his life in Woods Hole. Then he decided that if he wanted to be something that was going to be recognized for his science, he had to move to Europe. And on his 16th summertime, he decided to move to Stazione Zoologica Napoli. And that's there he became an important man. In fact, he moved first to Germany, to a university in Germany, but then he moved to Stazione Zoologica Napoli, and he began going there for 7 years every summer to Stazione Zoologica Napoli. He had a very bad time when he was going back to the United States. He didn't receive money for research, but he managed still to travel every year. And in the last 3 years, with more problems to come to Europe, he settled a little bit in Paris, in Sorbonne University. And being in Sorbonne University, he was for 3 years visiting Station Biologique de Roscoff. And it was in this last third year that the war was initiated in Europe, and Germans reached Brittany in 1939, and he was working there. He was set to leave, but he stayed in the lab until the Germans came, and he was made prisoner. By the time he had been married to a German lady, and the family of his wife managed to get him out from the concentration camp where he was. And when he was set free, he traveled to United States, and he died shortly after he arrived there. He was one of the most important embryologists in history. He's not very well remembered, but it's possibly the black man, the embryologist, a black man, most important that history has given to us.
Well, thank you for sharing that story with us. Ernest Just, right?
Yeah.
Right.
The Black Apollo of Science.
Apollo of Science. Okay, so back with the Challenger expedition, they've discovered some of the deepest points of Earth, and that there was life in them, like the Mariana Trench. Nowadays, we're finding plastic in these same places. Science has changed a lot since the early stages. So we would like to know if there's anything you envy from the lifestyle of those early scientists and their discoveries.
You envy many things. I mean, it's now 1 month that I'm telling everybody that I need to retire. I'm not very fond of computers, you know. I mean, I use a lot of computers because I work in transcriptomics, but I manage with the tools that I need for my research. All the things around my normal life in university and having to sign everything electronically and those things, I do not like it too much. I was in a lecture yesterday on artificial intelligence because I feel that I need to know something in order to understand what my students are doing with the assignments that I ask from them. And really, I want to retire. I'm very envious of those times in which you use your pen and your notebook, and you were doing your research like that. And I know I like movies like Indiana Jones. I would have loved to be born in the 19th century, but I mean that's not possible.
What if you had a whip too?
Yeah.
A whip?
Possibly not.
Possibly not. Right. Well, coming back to the present, what are the great marine expeditions going on in the 21st century? And is the marine station of Plencia involved in any way?
Yeah. Okay. With the upwelling of all this sequencing approach, we are able to sequence the world now. So you just need 1 milliliter of water, filter what is in there, and you can sequence the DNAs that is present there to know which microorganisms are living in 1 milliliter of water. So when the human genome was sequenced in the and this was presented to the world, I mean, you must think that this was an eureka moment in our history. So everything was presented in the White House. So it was the President of the United States of America with 2 scientists besides him, telling everybody that they have cracked the code of life. I mean, they had read the human book. And at that moment, I mean, the technology suddenly, it was like a burst in technology, and we were able to sequence very fast, very quick and cheap. And one of the men that appeared next to President Clinton was Craig Venter, and he's a sailor. He loves sailing. And after he published the genome of the human, he decided to take his boat and circumnavigate the planet. That was the Sorcerer expedition. And he was taking water samples and sequencing what it was present. That was was amazing. I mean, the amount of sequences that he was able to contribute to the gene bank was incredible. I mean, in just one paper, he contributed as much sequence information that as it had been contributed in history by all scientists. That was just in one paper. And yeah, the Sorcerer expedition, I mean, for me, it launched the 21st century. That happened in between the year 2001 and it finished in year 2007. And then there was another circumnavigation experiment, and you have had under this microphone one of the members of this expedition. Belén was in the Malaspina expedition, and it was also it was initiated as a circumnavigation expedition because the Malaspina expedition that it happened in the 17th century, that was a circumnavigation voyage, but this was not in the end the circumnavigation voyage, the one of Malaspina, but they were traveling in both hemispheres and mainly concentrating their research on deep waters. And they were also following the same approach, taking water and sequencing what is in there. And then in the 2010s, then in France, there was a new foundation. There's the Tara Foundation, that they obtained a boat, a sailing boat, and since the year 2010, they have dedicated themselves to navigate different waters and characterize molecularly the plankton that is living in the waters. And the amount of information that we have obtained from that, it has been amazing. So those have been, for me, Sorcerer, Malaspina, and Tara expeditions have been something very important. But then there is a very important research organization in Europe, is EMBL, the European Molecular Biology Lab. Myself, I was trained in Germany. 50% of my PhD happened in Heidelberg in Germany. I was in the University of Heidelberg, which is the oldest university in Germany. It's a small city. It's the size of Bilbao, and now it has got more than 10 Nobel Prize winners. And some of those prize winners, the last ones, come from the EMBL. And EMBL, those are international laboratories. They do not belong to Germany. The researchers coming there are from different places in the world. The money for this institution comes from the different member countries, most of the European countries. Spain is a member country. And they do very advanced molecular biology research and microscopical research in the lab. So this was one of the important institutions in research of COVID, for instance. Okay, but suddenly they have discovered that there is a world out there. I mean, there is a planet out there, and they work with executive programs for 5 years, and the one that have now is called planetary biology. And within planetary biology, they discovered that they wanted to study the coastal ecosystems of Europe, and they launched an expedition, which is the Trek expedition, traversing European coastlines. And in going to the coastlines, their approach has been, okay, we need a vessel that is going to be able to sample in the sea, close to the coastline, but in the sea, and which better which better partner to call than Tara. And they called Tara Foundation, and they incorporated the Tara Foundation for the expedition. Okay, when we go to the coastline, we need also to sample the intertidal environment. And we have very nice equipment in our laboratories, but we need to move it to the coastlines. So they came with the idea of mounting or arranging a big truck which is 40 meters long, where they have put very sexy equipment, the kind of equipment that, it encompasses 70 of those marine stations. And we are a member of EMBRC. And that's how the Trek expedition contacted us to make the Plencia Marine Station and the Bilbao Plencia environment a super site, because not all the places where they have been going for sampling have been super sites. There are some places in which they have gone, they have sampled, and they have moved on. Here they were put for, the track was for 1 month, plus 1 week of the Tara in the Maritime Museum of Bilbao. So we were one of the very sexy trek points in the expedition, and it was a fantastic thing. There were scientists from all around Europe coming to here, people that I know from the papers, and they were here, and you were able to touch them and talk with them and doing research with them. It was a fantastic experience. Now the Trek expedition is coming to the end. They are now they are in Rome at the moment, the truck, and the truck is going back to headquarters in Heidelberg, and Tara will be finishing the sampling in Greece. Now in June it finishes. It has been a 2 years expedition, and we have been a stopping point for more than 1 month, and hopefully the collaborations will continue with them.
What are the results obtained during those 2 years expected to?
The thing is that the depth of information that they are willing to obtain, it's enormous. You must think that the first circumnavigations of the Tara finished in the year 2013, and finishing in the year 2013, this year, last year 2023, 10 years after, there were still many papers coming out. So still we have a period for this information to come out. Just imagine, when they came here to Bilbao, they were coming from La Tremblade, from close to Bordeaux. And when they arrived in Bilbao, they stopped in Bilbao because Tara was not stopping in all the sampling places. They stopped in Bilbao, and they brought out from the boat all the samples collected during that month before coming to us. And all these samples were sent by truck to Heidelberg. And from Heidelberg, they had to be diverted. Some of the samples have to be sent to France to be sequenced in Genoscope. These sequences are still coming out, and when they are coming out, I'm talking about terabytes of information. And those terabytes of information have to be processed and analyzed. So still, the digestion of all the information that is coming from there, it will require a couple of years.
Have to be aware, at least for the following decade, no? For those results.
Yeah, I think it's going to be something else, because it's like an atlas of Europe, an atlas of Europe in the 21st century. If you imagine the Beagle expedition that was a geographic expedition to lay maps of South America, and so we are in the 21st century, and we are laying the first map of the microbial biodiversity in Europe. That's going to be something else.
So you've mentioned the magnitude those projects they have, and that's like playing in the Champions League.
Yeah.
And the PA has been able to play in that Champions League. Considering that the Plencia Marine Station is a rather young marine station, how did you manage to gain the reputation and be part of that?
Okay. Plencia Marine Station is young, but as a research group, we are not that young. I'm talking about retirement, so imagine we have been doing research for quite a long time. Jonan us, and we have some kind of a reputation in the field of environmental toxicology. In saying that, talking about environmental toxicology, it's quite a reduced group of scientists. When I'm talking about Trek, you are talking about the Champions League. We are talking about people that are doing high research in biomedicine, for instance, in ecology, but also in biomedicine. How we came to light to these people. Okay, when we opened doors in 2012, shortly after we organized an international congress. It was the ESCPB Congress, European Society for Comparative Biochemistry and Physiology, and we invited a scientist, Hans Otto Porna, from the Abbey in Germany, in Cuxhaven. And when he came and visited the marine station, he said, "Okay, this is a fantastic facility. I mean, it has been born to be part of EMBRC." EMBRC was in the moment in which it was being assembled. They were looking for different partners in different countries, and Spain was not in the roadmap. So we did knock at the door of EMBRC, and we said, "Okay, we would like to participate." They said, "Okay, because we can provide access to marine waters in the Gulf of Biscay." And this, okay, they say, "Okay, it's fine. If you obtain the okay of the Spanish Ministry of Science, you can come in." And it was funny because we did obtain this okay from the ministry very, very fast. They said, "Okay, we are not giving you any money." And we said, "Okay, we have the money. We have the money from the local government here in the Basque Country." And so the Spanish government was very happy if they didn't have to spend a single euro in us becoming members of EMBSC. So we became members of EMBSC. And becoming members of EMBSC is not becoming members of anything. For instance, in Italy, the main institution is Stazione Zoologica Napoli. Stazione Zoologica Napoli opened doors in 1872, 152 years. And there have been 21 Nobel Prize winners doing research in Stazione Zoologica Napoli. 21 Nobel Prize winners. 20 in sciences, 1 in peace. That's not anything. Another organization, it's Station Biologique de Roscoff, that opened doors on the very same year, 1872. And at least 3 Nobel Prize winners have been working there. Loeff and Wolf were working there. The fathers of the molecular biology dogma, they were working there as young researchers. 152 years of doing research. PML, the Plymouth Marine Lab, at the moment in which we joined, was a part of EMBSC. 12 Nobel Prize winners working there. And there are institutions that have not received Nobel Prize, but I mean, they are the hubs in which many other things happen. Bliss in Belgium, it's where EMOTNet is housed, for instance. So we are playing with the very important guys out there. Okay, we are young and we cannot do certain things, but we are meeting these people. And when you are meeting these people, the people sees that you are there. And you are in the window screen, and being in the window screen. I mean, I would never say that EMBL, the European Molecular Biology Lab, came here because we are the Plentia Marine Station. They came here because we are members of EMBRC. Yeah, but yeah, we did our best to become members of EMBRC, the European Marine Biological Resources Center.
Since we have the chance to have you here with us, we would like to know what advice or tips can you give us to help us in our professional careers as sexy scientists?
Okay, that's the very difficult part. I'm also a father, and I have a son who is 19 years old and who told me 3 years ago that university is for his father. He doesn't want to study anymore, and he likes the sea, and he's going to become a fisherman. So very possibly I will be saying goodbye to him when he's going to Seychelles to fish for tuna. What I'm telling you, ever since I was 5 years old, I knew that I wanted to be a biologist. I mean, that has been my driving force. I mean, everyone, it's everyone. And each one has got its own focuses and goals. And my idea was doing research. I always wanted to do research. But research is not for everybody. There are people that are doing the masters. because they want to dedicate their lives to teaching, for instance. And I cannot say that it's not a good thing, because I love teaching. I really enjoy being able to show others what I could know of things. But science is not for everybody, that's for sure. There are people that get very frustrated when they are in the lab, and even the people that see their horizon in doing science, sometimes after 1 year in the lab, they could be feeling that it's not for them. Okay, if you are really willing to do research, you have to pursue that career. There will be moments in which you'll be feeling very, very down, and it's a little bit a life of a monk or a nun, no, because it requires a lot of effort. But in my case, it's the thing that I like doing the most. I mean, I like staying with my family. I am lucky because my wife is also a biologist, so I can talk about biology with her. But I even do that with my mother, who is not a scientist. No, it requires an effort, and if you require the effort, try to look for those things that you like with the people that you could be interested in working with. Nobody needs to suffer. And in science, many times you suffer not due to the research work that you are doing, but because of the people you get involved with. So always try to work with friends. And that means in your PhD, in your work in your lab, but also in your collaborations abroad. The best thing of collaborating abroad is that you have the possibility to choose who you want to collaborate with. And maybe the wisest thing is not always collaborating with the one doing rocket science the whole time if it's a nasty person. Yeah? Collaborate with nice people. This is like making friends.
Fantastic. Thank you for that honesty. It's a very interesting answer. Well, we will also like to know what book or scientific paper or both you would like to recommend to our audience.
Okay, I should be recommending you a book that is not published yet, because I'm finishing writing a coffee table book on the history of marine biological stations. I mean, it should have been published 3 years ago, but my life is like it is. But we are now in the process of editing the book, and hopefully by the end of the year it will be published. It's a coffee table book. When I say coffee table book, it's the kind of book with many pictures, many photographs, and that you should be reading by the fire with a cup of coffee, or in my case, with a glass of whiskey. So that's the first book. I'm not going to be recommending The Origin of a Species, because for most of the people it will be a boring book. But possibly people do not know whether, I mean, I like science, but I like literature. And I do try to use it in my lectures. So if I have to recommend a book, I always recommend 100 Years of Solitude.
Well, Ibon, we haven't had the time to mention anything about your lectures, but I must say that they are fantastic, and the creativity there is behind those lectures does really have people completely involved, and you cannot not pay attention in your lectures. There you go, they're very energetic. So
I try to cut your guts.
There you go, absolutely.
You can't fall asleep, at least.
You can't fall asleep, it's impossible. So hopefully next generations of MER and from the University of the Basque Country will get to listen to this and get to see your lectures and everything. It's been fantastic having you here. Hopefully you will be maybe the first Nobel Prize in the pie.
I don't think so.
Or the tutor of one, maybe.
Yeah.
Or the tutor of one. That would be
my goal.
That would be, and maybe, probably possible. So thank you for being here with us. I hope you've enjoyed. And
thanks to you, and may the science be with you.
May the science be with us. Ibon Cancio, thank you.